Hi everyone, welcome to MR Learning Circle. I hope everyone is fit and fine and doing good in their lives. So today is the fourth lecture of our MCQ series and the topic today we are going to cover is Methods in Microbiology Part 2. And I hope you all have watched the previous 3 lectures. If not, then please go and watch it. It's really informative and very important for your upcoming GATE examination. So let's start. So today we are going to cover, the first topic that we are going to cover is enrichment culture technique for isolation of microorganism. And the first question is, the principle of enrichment culture technique is A. Increasing microbial diversity in culture. B. Providing conditions that favor growth of desired organisms over others. C. Killing unwanted microbes using antibiotics. And D. Isolating only aerobic organism. So the correct answer for this question is option B. That is providing condition that favor growth of desired organism over others. So here the enrichment culture, it provides the specific nutrients or environmental conditions selectively so that the growth of a particular target organism is promoted. Next is question 2. For isolation of azotobacter, the enrichment media should A. Contain nitrate or ammonia as nitrogen source. B. Be nitrogen free to favor atmospheric nitrogen fixation. C. Contain antibiotics to suppress non-nitrogen fixers. And D. Be acidic to inhibit fungi. So the correct answer for this question is option B. That is, be nitrogen free to favor atmospheric nitrogen fixation. So here what azotobacter does, it grows in nitrogen free media so that it could fix the atmospheric nitrogen, whereas other bacteria, they fail to grow. Now question number 3. Enrichment for sulfate reducing bacteria requires A. Sulfate as electron donor and oxygen as acceptor. B. Sulfate as electron acceptor and organic acid as donor under anaerobic conditions. C. Sulfuric acid as carbon source. D. Sulfide as nitrogen source. So the correct answer for this question is option B. Sulfate as electron acceptor and organic acid as donor under anaerobic condition. Okay, so here the sulfate reducers, they use the sulfate as the terminal electron acceptor in anaerobic respiration, whereas with organic acid should be the donor. Now question 4. To isolate cellulose degrading bacteria, the enrichment media must contain A. Glucose and peptone, B. Cellulose as sole carbon source, C. Sulfate and nitrate salts only, D. Starch as carbon source. So the correct answer for this question is option B. Cellulose as sole carbon source. Next is question 5. The selective condition for thermophilic bacteria in enrichment is A. Low temperature and acidic pH. B. High temperature and neutral to slightly alkaline pH. C. High salt concentration and low temperature. D is low oxygen and low temperature. So the correct answer for this question is option B. High temperature and neutral to slightly alkaline pH. So thermophiles, they grow best at 50 to 70 degree neutral or slightly alkaline conditions. Now we have question 6. For enrichment of nitrifying bacteria, the media should contain A. Organic nitrogen compound, B. Inorganic ammonia or nitrate as energy source, C. Nitrogen free medium, and D. Glucose and yeast extract. So the correct answer for this question is option B. That is inorganic ammonia or nitrate as energy source. So here the nitrifiers are chemolithoautotrophs. So they use ammonia or nitrate as energy sources and carbon dioxide as carbon source. Now question 7. Purification of an organism from enrichment culture is achieved by A. Direct inoculation from sample. B. Successive subculturing on selective medium until uniform colonies appear. C. Addition of multiple nutrients. D. Decreasing incubation time. So the correct answer for this question is option B, successive subculturing on selective medium until uniform colonies appear. Question 8. Enrichment for methanogenic archaea requires A. Aerobic condition and glucose, B. Anaerobic condition with carbon dioxide and hydrogen as substrate, C. Aerobic condition with nitrate, D. Sulfur oxidation. So the correct answer for this question is option B. The correct answer for this question is option B. Anaerobic conditions are with carbon dioxide and hydrogen as substrate. Why? Because methanogens are strict anaerobe. Clear? Now question number 9. Enrichment cultures may not represent natural diversity because they favor fast-growing and condition-tolerant species. B. All microbes grow equally under enrichment. C. Enrichment increases microbial diversity. D. It isolates only virus. So the correct answer for this question is option A, that is they favor fast-growing and condition-tolerant species. So selective conditions, they favor the dominant or the fast-growing microbes. So what do they exclude? They exclude slow growers or minor population of the microbes. Question 10. The Vinogradsky column is an example of A. Differential culture technique, B. Enrichment culture showing microbial succession in a natural gradient, C. Pure batch culture, D. Continuous culture system. The correct answer for this question is enrichment culture showing microbial succession in a natural gradient. Now we have next topic, that is antigen and antibody detection method for microbial diagnosis. So first question from here is the principle of ELISA, that is enzyme linked immunosorbent assay, is A. Precipitation of antigen in solution, binding of antigen or antibody to a solid surface and detection using enzyme label antibodies. C. Agglutination of RBC by antibodies. D. Complement mediated lysis. So the correct answer for this question is option B, binding of antigen or antibody to a solid surface and detection using enzyme labeled antibodies. Important question. So question number 2. In a direct ELISA, what is coated on the solid phase? Options are A. Enzyme labeled antibody, B. Antigen, C. Secondary antibody, and D. Complement. So the correct answer for this question is option B, that is antigen. So in direct ELISA, the target antigen is immobilized on the plate and enzyme labeled antibody binds directly to it. Now question number 3. In indirect ELISA, the signal is amplified because A. Enzyme acts on substrate, B. Secondary antibody binds multiple primary antibodies, C. Complement binds antigen, and D. Agglutination occurs. So the correct answer for this question is option B, secondary antibody binds multiple primary antibodies. So this is basically the principle of indirect ELISA. Question 4. The principle of Western blotting is A. Separation of protein by SDH page followed by detection using specific antibodies, B. Precipitation of antigen in agar, C. Agglutination of red blood cell, D. Complement fixation. So the correct answer for this question is option A. Separation of protein by SDH page followed by detection using specific antibodies. Question number 5. Agglutination test detects option A. Soluble antigen only, B. Soluble antibodies only, C. Interaction between particulate antigen and antibody causing visible clumping, D. Enzyme activity. So the correct answer for this question is option C. Interaction between particulate antigen and antibody causing visible clumping. So in agglutination, it involves clumping of cells or particles coated with antigen when specific antibodies are present in that particular test. Now question number 6. Complement fixation test is used to detect Option A, free complement in serum. B, antigen antibody complexes that activate and consume complement. C, enzyme activity. D, microbial growth on media. So the correct answer for this question is also option B, antigen antibody complexes that activate and consume complement. Next question. Fluorescent antibody technique rely on A, enzyme labeled antibodies. B, fluorescent label antibodies binding specifically to antigen. C, agglutination of RBC. D, precipitation in agar gel. So the correct answer for this question is option B, fluorescent label antibodies binding specifically to antigen. Question 8. Radioimmunoassay RIA principle is A, antigen antibody binding detected using radioactive isotopes. B, enzyme activity detection. C, enzyme precipitation of antigen. D, hemolysis of RBC. So the correct answer for this question is option A, antigen antibody binding detected using radioactive isotopes. So these antigen and antibodies, they are radioactively labeled molecule and they are highly sensitive for low abundance target. Now question 9. Lateral flow assay, that is rapid test, detect A, antibodies in serum or antigen in clinical samples using label antibodies on nitrocellulose strip. B, complement activity. C, enzyme activity in microbes. D, microbial growth in agar. The correct answer for this question is option A, that is antibodies in serum or antigen in clinical samples using label antibodies on nitrocellulose strip. Question number 10. Precipitation reaction in agar gel is used to A, detect antibody antigen complexes forming visible precipitate. B, measure complement activity. C, detect enzyme activity. D, identify microbial growth. So the correct answer for this question is option A, detect antigen antibody complexes forming visible precipitate. So that was all for lecture number 4. I hope you find this lecture important, and if it is beneficial for you, please like, share, subscribe and do share it with your friends also. Thank you for watching.